Swayam Prabha. Digital India. Educated India.
Hello, welcome to Metals in Biology. Today's topic is photosynthesis. You will be amazed in knowing how great this reaction could be, where we will be converting water into oxygen. Now overall, if you see all the transformation, perhaps two reactions stands out. Of course, there are many reactions which stands out, including the methane to methanol. Perhaps two reactions which are complementary with respect to each other and they are very much fascinating, and that is oxygen to water formation as well as water to oxygen formation. Life comes to a circle, I would say. So, reduction of molecular oxygen to give the water and water oxidation to give you the oxygen, I think are the two most important processes that is keeping us alive. So, it is happening, this oxygen to water is happening at mitochondria, where you have seen the cytochrome C oxidase, the heme copper oxidase, and the intricate details of it, that you have seen how oxygen is converted to water by the heme copper oxidase, which is quite fascinating. This is the mitochondria where things are happening. The cytochrome C oxidase, heme copper oxidase is getting involved in there to convert oxygen into water. On the other hand, in chloroplast, in plants, we have this photosynthesis where you have photosystem 2, the oxygen evolving complex, we will see in a moment, manganese 4 calcium compound or the structure cluster will be involved in the water oxidation. So, water will be converted to oxygen at chloroplast over there. These are complementary reactions and these are right diagonally opposite reaction. These of course requires 4 proton and 4 electron for converting oxygen to water, and the reverse will be true for the water oxidation to make the oxygen molecule. These transformation require, both these transformation require multiple metal center. So, that is quite fascinating. You see the metal is everywhere, of course more than 50% of our body weight is metal, but more importantly wherever there is a difficult reaction or difficulty arising, nature has always trusted on metal. Metal is the place to go when you have seemingly nowhere to go. Metal makes the reaction feasible. Metal play multiple roles in these enzymes, both in this today's case let us say heme copper oxidase and the oxygen evolving complex chloroplast. Metal plays multiple roles such as it binds with substrate meaning in these cases let us say oxygen or water, increase reactivity of substrate, of course increase the reactivity of oxygen or water. In this case it would be water activity, water binding and water activity will be improved. Prevent side reaction, which is important and essential because these biological processes are completely bulletproof, right. These are the reactions which happens no matter what. This is one dimensional, single way, one product formation or the desired product formation only. There is no detour, almost no detour nowhere, and then almost no other side product formation happening. Of course, depending on the concentration of the materials, things can vary. In few cases that we have seen, for example peroxidase, catalase cases we have seen, but otherwise it always prevents side reactions. Provides electron quickly, that is one of the great thing about the metal center. Metal centers are used very cautiously and very carefully, particularly wherever required to provide the electron. It can couple a multi-electron process, that is also important. Of course, at a time only one electron transfer occur, but it can provide multi-electron process to several single electrons process. It can have multi-electron transfer at a time or several one electron transfer at a time or at times. So, you will see in a moment that this OEC, oxygen evolving complex, will have 4 manganese center. You have already seen in the previous classes where cytochrome C oxidase, you have iron copper site along with a phenol or tyrosine cross linking over there. So, these manganese, 4 manganese calcium 1 calcium structure we will see today for the oxygen evolving complex or for the photosynthesis where water is converted to oxygen. The diagonally opposite, exactly opposite, cytochrome C oxidase confirms transformation of oxygen to water. Of course, another very important enzyme is this nitrogenase, which can convert nitrogen to ammonia. These are fascinating enzymes, these are fascinating fascinating transformations. We will come back to that in a moment. Let us look at the crystal structure of the photosynthetic oxygen evolving center or oxygen evolving complex. This is back in 2004. This is the crystal structure that you see is reported. Many structures are present, as you can see, non-heme iron center, heme center and cytochrome center, many different centers are present, many heme and many non-heme centers are present. Many structures are present, like this sort of different structure is, as you see, the one in the green here is the one which is the photosystem 2, and this is a great structure for the photosystem 2. As you see, this is the oxygen evolving complex or oxygen evolving center, this is the chloroplast structure in green over there. Of course, this photosystem 2 dimer structure is existing, but it does not still provide the definitive structure of OEC. So, what happens here is this crystal structure, which is phenomenal and highly informative, but still it cannot give you the resolution at a level what a inorganic chemist would like to have perhaps, right. These crystal structure are fascinating, many different iron center, heme iron, non-heme iron center, many different metal centers are present. Despite the presence of these many different centers, we see that still it cannot give the clear idea about what is happening at these metal centers and what is the metal, what are the exact association of these metal centers. Okay. So, we will see this OEC, oxygen evolving complex, over here. In a moment we will be coming back, but what has been known or what has been understood so far is these 4 manganese 1 calcium structure, which will start at S0, we will come back to that in moment also. It is photosensitized to give you S1, then subsequently S1 can form S2. Once again these are the subsequent redox reaction, each of the step involve 1 electron processes. The catalytic cycle of OEC is right over here, it is going through S0 to S1, S1 to S2 and S2 to S3 and S3 to S4. So, all these steps are dependent on light, and it is in turn oxidizing metal oxo cluster. in each step metal is getting oxidized further. Okay. Finally, at S3 S4 the catalyst or 4 manganese center 1 calcium center is getting ready to react with water to give you the oxygen molecule. The S4 to S0, that means the final step where water is converted to oxygen, is light independent. This step is light independent and releases oxygen. This overall transformation is very complicated and intermediates all have been postulated, not direct evidence. I mean, some evidences are there that we will see in a moment, but these are not 100% clean and clear informations. But most importantly, as you know, by doing or by converting water into oxygen, this whole process is also coupled with the carbon dioxide to form the glucose, which is the pretty important molecule for our existence, right. Water to oxygen conversion is also coupled with carbon dioxide, that is, I think you are perhaps more familiar with carbon dioxide to the glucose formation. This whole process are coupled and we will see stepwise what perhaps is happening over there. I think one thing for sure, unlike many other enzymes that we have seen in this class, this enzyme is perhaps the most complex and very little understood. It is not, at least I mean it is understood well, but it is not up to the mark what we would like to have, because the thing is these are very sensitive metal cluster that is involved, that we will see in a moment. There are many propositions that what would be the relative disposition of these different manganese center, but no direct or no clear indication exists till that. Okay, there are proposals, there are argument in favor or disfavoring one of the structure, but nailing down one structure has still been inconclusive. I would say still lot of studies are going on. These are fascinating structures both in terms of the enzyme study as well as the synthetic studies has been done quite a lot. So, the possible arrangement of the 4 manganese ion that we are talking are many, and some of them are shown over here. As you can see, many of them are having the butterfly like structure, and these are the structure in the box which are getting support from the X-ups measurement. So, still from the X-ups also many structures are proposed. It is not really one definitive structure or one clear structure that can be proposed, but for you know the course purpose or keeping it simple, we will slowly evolve into one structure and try to say that let us try to follow that rather than trying to follow many structure. Let us keep one in mind, knowing very well that that structure may or may not be the correct one, because there are many structure. As you see, the different permutation and combination is possible between the 4 manganese and 1 calcium orientation, as at least if you are just focusing between 4 manganese 1, 2, 3, 4 manganese, you can have all these different structure orientation bridged between by the oxide, right. So, manganese and calcium also comes into the picture, how they are bridged, how they are helping, that can be debated. It is still debated, no clear solution once again exists for this beautiful enzyme and most effective, one of the most effective enzyme that is converting your water into oxygen, right. That is fantastic. We will try to see what is understood, what is evolving, and how we perhaps will be able to understand it better in future. So, let us get back to that crystal structure, that OEC crystal structure. 3 slides back we have seen this is the crystal structure. If you zoom down over here further, if you zoom down in this region, you see that these are 1, 2, 3, 4, these are the 4 manganese clusters that we were talking about. This is the calcium over there. So, what is the debate about? Well, although this is looking like a definitive structure, people do not believe that that is the original structure. What has happened during the crystallographic characterization or crystal data collection, people believe that this cluster is broken and rearranged. So, or I mean this is not actually is the real structure. So, X-ray damage to the manganese 4 calcium complexes has happened already, although this is a decent 3.5 resolution structure which allows the density modeling, but it is still not definitive. Okay. The mu oxo bridges that is present over there and the hydrogen bonding network are not really well resolved, and this remains one of the most debated structure, most debated active site so far. So, the structure perhaps that we can move along with is this one 1, 2, 3, 4, these are the 4 manganese bridged by these 4 oxo, and also it is shared by the calcium. These are the 2 references you can study which are quite informative in any case, but still the debate remain what would be these 4 manganese doing, and of course their role is little understand, but how they are relatively positioned. You see these as if like these are dimanganese dioxo structure. There are different way you can look at this structure. This is perhaps the one, if you want to remember something, perhaps this is the one from the crystallographic characterization. Let us look at little bit more into the electronic properties of these intermediate. So, you start with S0, of course all these intermediates are having 4 manganese 1 calcium structure. So, from the S0 which is the fully reduced form, it is getting oxidized at each step. Each of these 4 steps are dependent on light, but the final step is not dependent on the light, that is converting S4 into S0. This light dependent step oxidize the manganese core to give you a H1 intermediate which is oxidized by 1 electron. At every step 1 electron oxidation is happening. We just do not know or for sure what was the manganese 4 different manganese oxidation states are, but nonetheless these will be oxidized by 1 electron, this will be also oxidized by 1 electron, S1 to S2, S2 to S3 another electron goes out in presence of light, S3 to S4 another electron goes out. This is the fully oxidized form, this is the fully reduced form. The fully oxidized form now will react with water to give you oxygen and reforming the fully reduced from S0, and that is how the catalytic cycle would go like, right. You have seen the cluster, what perhaps it could be talk over here, although knowing very well that is not the right representation of the structure that one would like to have. So, let us look at the same thing once again. We are trying to understand where water is oxidized to oxygen, and the structure of the photosynthetic 4 manganese 1 calcium cluster from X-ray spectroscopy that has been done, and then what are the caveats remain. This is a fantastic paper to read if you are trying to understand, but let me tell you that many things remained unclear. These are some of the proposition, still many evidences are required to nail down it further. So, the S0 state, as you can see over here, these are the proposed structure or proposed oxidation state assignment by the EPR multiline spectrum, but it fits with both one manganese being 2, one is 3 and another being +4 or another 2 being +4. 2 plus 3 and +4, 2 of them is proposed, or alternatively it could be 3 of them is in +3 oxidation state and 1 of them in +4 oxidation state. This is the fully reduced form. From there on 1 electron oxidation occurs, and this is quite definitive, and that is that upon oxidation of one of these 2 intermediate, perhaps one of them these 2 are correct which have spectroscopic evidence, we see that it would be manganese 3, 2 of the manganese will be in +3 oxidation state and 2 of the manganese will be in +4 oxidation state. Okay. So, these gives the EPR and EPR very characteristic for these species. Further oxidation of this intermediate gives rise to a situation where once again one of the manganese remain in +3 oxidation state, another manganese which was in +3 oxidation state now oxidized to +4 oxidation state. So, 3 3 of the manganese is in +4 oxidation state, 1 of the manganese is in plus oxidation state. Once again this will give rise to the multiple EPR, multiple line spectrum with 2 different G values, one for the manganese 3 plus and another for manganese 4 plus. These are quite significant. Understanding what we have from S2 to S3, then it is further oxidation. That means we have a all manganese 4 plus oxidation state or a radical intermediate where manganese 3 and manganese 1 manganese 3 plus and 3 of the manganese is in +4 state as well as a radical formation. Okay. So, these are the EPR spectra study. Now the assignment of oxygen evolving complex or center electronic structure is complicated and controversial. Okay. Let me get to that once again, that these are putative assignment. Perhaps some of them are correct, but definitely not all of them are correct, and there are open discussions on this, there are open debate on these still ongoing in the literature. These different structure or the different oxidation state assignment was possible by various EPR studies. These are complex EPR studies, one has to be really expert in EPR to study and conclude or try to propose some of the valid oxidation state from these EPR studies. FTIR studies, XS, JAS studies and UV visible studies were conducted for these to give rise to the original proposal that we have seen over here. So, finally, once you have these S3 state, one more electron transfer, one more electron oxidation and subsequent water to oxygen formation is going on. We will see these in a ChemDraw format real soon. Okay. So, what we have seen so far then that is saying that perhaps there is a manganese 5 oxo formation. As you see, if you just for simplicity pick up only one of them, let us say you pick up this one. So, 4 manganese 4 plus, all of them are 4 plus. So, further 1 electron oxidation has to have at least 1 electron oxidized to manganese 5 plus. So, this perhaps will have 1 manganese 5 plus. Okay. Other 3 manganese could be in manganese 4 plus. So, you can remember one of these oxidation state in each of them. So, although these two are possibilities, these two are possibilities, but S4 will definitely have a manganese 5 plus. So, that manganese 5 plus oxo would be quite electrophilic in nature, and quite naturally calcium hydroxide, which is generated from aqua or water molecule, can attack on this as a nucleophile is attacking on the electrophile to form the oxygen bond. So, this is the most critical bond we are looking for, because water to oxygen formation would require not the breaking of the bond, but the formation of the bond, and this is where perhaps that oxygen bond formation is happening. This is the terminal manganese 5 oxo species we are talking about. We will come back that to in a moment, but it could be also the bridging oxo, that bridge between the 3 manganese center can also be possibly forming the oxygen bond. These are the structure, or of course different orientation would be possible. Instead of manganese 5 oxo in this format, one can have manganese 4 O dot radical, where OH will be attacking the oxygen. Calcium hydroxide is also will be attacking the oxygen in these cases. Okay. So, this is what we have come to. This is again a simplified one of the cluster we are picking up, not every cluster we are talking about, one of the cluster which is proposed one, but it could be very well be wrong, but for simplicity let us try to remember this, where we are saying that 2 of the manganese center are always is in +4 oxidation state, that although we did not say like that over here, let us say we are ruling out this as a possibility. We are just for these are still argumentative, we are just for clarity just taking this one. 2 manganese is in +4 oxidation state and another 2 is in manganese 2 and manganese 3 oxidation state. What we see over here, these center will remain constant, nothing will happen to this structure throughout the catalytic cycle, or this is again a presumption, not direct evidence are existing, but it is becoming simpler if we are trying to think like that. 2 manganese center is in +4 oxidation state bridged by oxo, just like bis mu oxo intermediate. What you have seen in the last class in case of let us say intermediate Q in that your methane monooxygenase cases, right. This was iron 4 oxo. This is now a constant site or the site which is not involved into the oxygen bond formation. It is linked with another dimanganese center, which is of course it will be setting the redox potential really correctly. In any case, this is bridged by another oxo molecule, and from there you have a 1 manganese 2 and 1 manganese 3 center. It is proposed that in this case you will see one of the water molecule will be bound with the manganese, and therefore this calcium hydroxide which is right over there will be then subsequently interacting in the future cases. So, in the next class we will be discussing the details of these steps, how this stepwise mechanism is giving you water to oxygen, but it is important to understand that this once again the mechanisms are not really crystal clear. We really need to do much more studies to better understand this OEC or the photosystem too. One thing is for sure, these are 4 manganese 1 calcium cluster, and these are going to be the most fascinating and perhaps the longest unsolved enzyme so far, where we despite having the crystal structure still we are not sure what is going on. We will see how the oxygen bond formation is happening in the next class. Keep studying photosystem, we will be back soon.